Pam Dalton, talk about her views. Her biography is in your handout, and I'll just defer to her.
Thank you, Craig. And I thank the organizers for inviting me here today. I've participated in some biocycle meetings in the past. It's been a while, but it's good to be back among people who appreciate the kind of science that we do at the Monell Center, which is to try to understand the human response to odors in the environment. So obviously we all know that people complain about odors, particularly when they think a neighboring facility is generating them. And increasingly we see a lot of litigation. I mean, yesterday I spent my day walking around a hog farm in Missouri with excellent anaerobic digestion going on in the lagoons, I might add, but nevertheless, there's always a neighbor that is trying to bring a lawsuit against these types of facilities. And sometimes it can get very, very contentious. You face a unique problem in the compost and anaerobic digestion. Many years ago, I was asked by the Department of Defense to try to find a universal malodor. And what we came up with was a combination of different biological odors of decomposition. But as part of this, there was some publicity on the project, and I started getting helpful comments and hints from hundreds, if not thousands, of people about what was the world's worst odor, in their opinion. And here's the top three. And basically, except for the decomposing bodies, this is what you folks are dealing with, is rotting food, decomposing food, and to some degree, waste products. And these are odors that not only are considered unpleasant, but they also provide some warning information to people, which is probably why they're so salient in terms of their perception and response. Now, it's interesting. We don't have any direct information, unlike the taste system, where we are hardwired as mammals to prefer sweet things and reject bitter or sour at a very young age. It appears that most of our responses to odors are actually learned, not innate. And so that also leads to the amount of diversity and the variation in the population you see as to what is a bad odor. I've met people, for example, who really enjoy the odor of skunk, and other people who think that's the worst odor in the world. And it really has a lot to do with their experience when they first experienced that odor. If they were walking in the forest and they were having a wonderful time and they smelled a skunk odor, that may become a positive association for them. So here we have some examples of malodorants. And as Craig went on, many of these are able to be detected at extremely low concentrations. But there's also a lot of diversity in whether people find these to be malodorous or not. And I'll talk a little bit in my presentation about the sources of variation in the population that we study to try to understand how people perceive and respond to odor. So we think what makes an odorant a malodorant is basically these are mostly compounds that arise from the decomposition of organic matter. And as a consequence, when you think about it, that can provide a danger signal. I mean, you want to know if the food that you have stored in your refrigerator is spoiled or not. As well, you would also want to know if there is a decomposing corpse in your environment. So essentially, the olfactory system was evolved as a warning system, something to tell us when there's something in our environment that we should be aware of. And so this probably forms the basis for the class of malodorants. Some of these malodorants appear to transcend culture. We found that out in the DOD investigation, where we found that odors of human waste, for example, were repellent to everybody, no matter what part of the world they came from. And again, this seems to stem from this primitive emotional response that we have to the warning signal that the odor actually provides. Now, odors are uniquely emotionally arousing, and that has to do with the way the olfactory system is wired in the brain. it turns out that the olfactory system is part of the limbic system, which is part of our emotional response. Okay, so we have a structure in the brain called the amygdala, which when we are emotionally aroused, lights up. This is very, very tightly associated with the way odors are processed in the brain. So perhaps more than visual stimuli or auditory stimuli or tactile stimuli, olfaction has some privileged access to activating these emotional centers in our brain, both positively and negatively. However, the negative response is usually much more robust than the positive response. And it turns out that historically, people thought that the smell of bad odors was actually how disease was transmitted before we understood germ theory. So in other words, in places where there was so-called bad air or miasma, they would think that if they got rid of the bad odors, then the ability of getting sick in that particular environment would be reduced. So this was a very common way of thinking about it. And in fact, I think we still have some of those feelings, sort of a naive way of feeling about an environment. When something smells bad, we think it can make us sick. And I'll talk a little bit about that as well. So now you've heard about the different sources of odors, either in the materials as they're stored or in the products that are formed, either into biogas or land-applied biosolids. But what are actually the biggest problems? Well, I just did a Google search for anaerobic digestion plant odor issues, and I got 345,000 hits on it. So clearly there's a lot of information out there on this, whether it's blogs or reports in the news and that sort of thing. Some of the popular press said that residents report stomach-wrenching smells. They're made nauseous. They can't enjoy their properties. They can't keep their windows open. And as you all know, odor is critical in being the big driver behind getting plants permitted or actually reducing their operations, or in some cases, shutting them down completely. But you're not alone. I mean, it's not just compost and AD odors. I mean, here's something from the New York Times about a couple of years ago in Austin, Texas, where people were complaining about barbecue odors smelling all the time. So it's not simply just the quality of the odor. It really is a loss of control over their environment that they're reacting to. And even more unusual is in states where marijuana is now legal, people are complaining about the smell of cannabis. So there intentionally is the feeling that if an odor invades your home, your property, whatever, you have lost control over that. And that is also a big driver in why, whether the odor is a true malodorant or not, people complain to both regulatory authorities as well as calling up legal proxies. So there are a lot of challenges for your industry. And increasingly, nuisance odor lawsuits are being brought by neighbors, and in some cases, not necessarily being won, but being forcing these operators or these companies to settle with them over operating hours, the amount of material that they can handle, as well as other things like that. Now, as you've heard, there are some ways of measuring odor that the regulators do in some cases understand. So these so-called permissible odor limits at the fence line are often expressed in the dilution to threshold ratio. And some of the limits, if you look across the states or the municipalities that use these, you'll find dilutions to threshold ranging from a 4 to about a 7. And like, for example, in Missouri, it's a 7-to-1 dilution to threshold off the fence line that actually will activate a violation. But really, there's no uniform regulation across the country, across states. It's really a very patchwork system of trying to come up with a way of regulating odor. And that's very unsatisfying to many people, but it's where the industry, where the state of the science is at this point in time. In fact, many municipalities simply regulate or issue violations based on the number of complaints that they get from neighboring communities. And when you get a small group of activists who will form a telephone chain or an email chain where they smell an odor and they'll alert everyone else, you can get a lot of violations in a short amount of time based on that kind of regulatory approach. So I'm going to talk a little bit about how we perceive odors and how we respond to odors and all the variation that exists. So you heard about the fact that we have volatile chemicals. Obviously, if we're going to smell it, it has to be volatile. And we have to breathe them into our nose because the part of the nose that actually perceives odor is located way up here, right behind this cribriform plate. And so about 10% of what's available in the airstream is what makes it to these olfactory receptors in the epithelium. And once they do that, the molecules bind to these receptors, sort of the lock and key model, where the molecular keys fit in these olfactory receptor locks, and when they bind to the lock, then the receptor sends a signal to the brain that triggers our sense of smell. And so we have about 400, humans have about 400 receptors. Now, some of them don't work. Some of them are what we call orphan receptors, probably worked maybe hundreds of years ago in mankind, but are no longer relevant, and so they're no longer operable. And in fact, we don't really even know what the relationship is between the ligand, the chemical molecule, and the odorant receptor in many cases. Only a very few of them have been adequately characterized. But basically, the way the system works is that any chemical appears to be able to activate more than one type of receptor, and any single receptor type will also respond to more than one chemical. So it's the pattern of activation across the receptors that actually allows us to distinguish something like cherries from the smell of rain, petrichor, when rain hits the ground and you get that very distinctive smell. We call our sense of smell primitive, not necessarily because it's a simple system. It's not. It's actually far more complex than the visual system or the auditory system. But it's primitive in that in phylogenetic terms, it actually evolved very early. It was the first sensory system in the smallest of organisms that was capable of detecting chemicals from a distance. So obviously this started in an aqueous, a water medium, but it would allow an organism to move towards a food source or away from something dangerous. And so in that sense, it's primitive. It's part of the old brain, and that is why it's also wired to the emotional system. And it turns out that a significant portion of the human genome is actually devoted to encoding the receptors that allow us to smell. Now, we thought that was pretty unique and pretty amazing. However, it turns out that these olfactory receptors don't just exist in our nose, they exist throughout the body. What their function is in other places, for example, you can find them in male testes, and they probably help sperm find, their way through an environment. But we just don't know all of the functions of olfactory receptors yet. But today I'm just going to concentrate on the ones in our nose because that's where it's relevant for odor perception. But it's good to know that we really have a lot of the genome that's devoted to encoding and producing these odor receptors. So smell does differ from our other senses in some very important ways. It seems to have played a very key role in human adaptation and survival. As I said, the olfactory areas of our cortex are much older than those of other senses. And contact between the external environment and the brain is more direct with olfaction. So signals that are triggered by other senses, vision, audition, touch, are processed through the thalamus, which serves as a gatekeeper to those other parts of the cortex that actually process those signals. But signals from smell directly go to the olfactory bulb without this thalamic delay, which is probably why when you smell something, you react almost emotionally before you even recognize or be able to identify what you're actually smelling. It's a very direct signal that goes right through the limbic system and then to the olfactory cortex. Now, we often think of our sense of smell as being not as good as many other mammalian creatures. And of course, some of that has to do with the hardware. We have fewer olfactory receptors than our canine friends, for example. But it turns out that our sense of smell is better than we think it is, and that often causes us to underestimate or overestimate how much chemical is in the air when we can smell something at the part per billion level or even the part per trillion level. We think because we smell it, that means there's a lot of chemical available and we're being exposed to something that's unacceptable, but in fact we're very good. This was a study done by a colleague of mine who wanted to see if people could track a scent through space. So here what you see is sort of the, let me see if I can find the, yeah, there it is. Okay, so this is the way a dog would track finding a bird. You can see this is the straight line that the bird fell when it came down, but the dog is going back and forth, and when it loses the scent, it moves back towards the scent again, and when it reaches it, it continues on that track. So what he did is he took a group of undergraduates on the Berkeley campus, and he took away their sense of sight, and actually had them follow a trail of chocolate across the grass. And as you can see from the tracking here, the person did actually very well, even on their first attempt. And later, with practice, they got very good at it. They were very, very tight keeping that line. So in many respects, we do use our sense of smell in ways that we don't appreciate. Exactly. Well, I guess if they could have been told anything, they might have been motivated. They were also paid to do the experiments, so that might have helped a little bit. So the bottom line, these relevant features that I think are important for understanding odor complaints is that we're much more sensitive than we believe we are, and we can smell at the pp parts per billion or even parts per trillion level for many chemicals. Olfactory stimulation is inherently emotionally arousing. We smell an odor and we get this sense of what is it, should I move towards it, should I move away from it? The system is optimized to detect very tiny differences, but we have very little ability to discern the safety or the hazard from the way something smells, and even less confidence in our ability to identify the source. So this causes a lot of people to actually overreact to odors that they may detect. It did evolve as a warning system, and taken together, all of these features can lead people to become anxious when they smell an odor, even when it's of no toxicological concern for them. So as Craig went through, there are a number of different ways of characterizing different thresholds. My interest in studying is usually between discriminating the odor detection threshold from something that we call the sensory irritation threshold, because that's actually transduced by a different system in the, let's say in the airways, the trigeminal system that reacts when any volatile chemical gets to a certain concentration. There are almost few examples that you cannot reach an irritation level. So what I'm talking about here is the concentration of the chemical. At a very low concentration, some people in the population will be able to smell it, but eventually you will get 100% of the people who have a reasonably normal sense of smell to detect it when the concentration gets strong enough. But over here, you have what I'm calling the irritation threshold, which is usually an order of magnitude or more higher than the odor perception threshold. Now, why is that relevant? Well, it's relevant because people will often say that the odor makes them irritated. It causes their eyes to burn, their throat. They'll complain about these irritation symptoms. But here I've just plotted what the odor threshold is for a number of compounds that, that might be detected around a facility, and the corresponding irritation threshold. Now, there is no irritation threshold for H2S. It's actually not a sensory irritant at any concentration that could be tolerated. It does not produce activation of the trigeminal system. But these other, for example, here you can see the concentration of dimethyl disulfide, and yet the irritation threshold is far, far higher. So when people start complaining about irritation from the odors that they experience, in general, what they're talking about is a psychological reaction, not a true physical sensory irritation, because it's unlikely that off-site you would ever see concentrations in the community reaching the levels necessary to actually elicit true sensory irritation. And it's also the case that when people smell bad odors, they can elicit stress-mediated symptoms, such as headaches, for example, or nausea. This annoyance or disgust about an odor and the consequent loss of control can lead to people reporting a constellation of different symptoms. It's also the case, when I said odors are emotionally arousing, that just the awareness of the presence of an odor can make people think about how they feel. Maybe they had a headache before they ever smelled the odor, but now they're going to attribute the headache to the odor or whatever other symptom they're experiencing. And when we talk about people coming out to evaluate this, we have to remember that the community that has gotten energized or aroused or, let's say, active about a facility, they may be experiencing more stress when they experience an odor than somebody who works for the facility and who's just been sent out to understand what the level or the intensity of the odor is. So why do people experience odors differently? What's the source of variation? Well, it turns out that it looks like we all live in our own different olfactory worlds. And the problem with that is that there's no good way to compare that. We don't have good language for describing what we're smelling in many cases. And all we can do is reference a source. You know, it smells like oranges or it smells like pizza. And that's not good enough because, like someone who's colorblind, they may not see red and green in the same way we do, but they've learned to call out what is a red color based on their experience. So what you may call pizza or what you may call the smell of, a compost facility could be very different quality-wise. Now, why is that? Well, first of all, there's a lot of genetic variation in how we express odor receptors. And so I said there are 400 different types of odor receptors, and some are non-functional. But some people fail to even express odor receptors and still have what we would consider a normal sense of smell. There's also considerable variation in the anatomy inside your nose, which, remember I said 10% of what you breathe in makes it to the receptors? Well, the positions of the bony turbinates in your nose and other things are going to make that a little bit more or a little bit less. So there's another. Gender, age, and disease. Obviously, as we get older, most of our sensory systems decline, and olfaction is no exception to that. It's also the case, I'll talk a little bit about experience and exposure, that when we are exposed to odors chronically, we become desensitized to them, as well as we stop paying attention to them in a more habituation-like sense. And also the expectations that we have about smelling. So the genetic variation, basically, we call this specific anosmia. Anosmia is the inability to smell. Someone who is completely anosmic can't smell anything, either because they had a head injury or they were born that way. But specific anosmia is probably relevant to all of us. If we were able to test your sense of smell on 1,000 different compounds, I guarantee you that everyone in this room would be specifically anosmic to one or more of them. There would be something that you couldn't smell because you didn't express that receptor. And so what does that mean? Well, we never really test people like that on that many odorant compounds. And it's not always relevant because most of what we smell are mixtures. But what that means here in this little schematic of the olfactory receptors that send their signal to the olfactory bulb in a very, very measured way, that if you remove some of those, what was smelling like a geranium might now just smell like citrus to you because that's part of the mixture of what a flower generates. And so you've removed some of the elements from that. So the person smells something, but they don't know how to characterize it. It's the same. So there's that quality difference across individuals based on their genetic variation in the expression of odor receptors. It's also the case that we're getting a lot more sensitive to odors. As we have reduced all of the background odors in our environment, we've done a very good job of that, at least here in the United States, is that we become more sensitive to the odors that are generated. So essentially, we have a signal noise distribution where now the background noise is so low, in other words, in terms of the odor landscape, that when we do smell an odor, let's say from a facility or an industry, it's much more noticeable than it once was. And it's also the case that publicity and the ability of social media to get people aroused about these things can also sensitize us to some of these emissions and product odors. Adaptation. Anytime anyone is exposed to an odor for even 30 seconds, your receptors stop responding in the same way, and you will find that that second or third or fourth sniff of an odor is much less intense than the first one. Think of it when you walk into your home and there's a good cooking odor around dinner time. Well, when you first open the door, it's very noticeable, and after 5 minutes, you're barely smelling it anymore unless you go into the kitchen and stand right over the pot. So this is relevant to Craig's point about why you need someone other than a facility personnel to evaluate the odor intensity or the presence of odors around a facility. It's not because they're biased, because they don't want to smell the odors. It's because they can't smell them. The receptors are no longer responding. And we have looked at this in a number of different circumstances. So here's a study I did a few years ago looking at long-term adaptation from simply exposing somebody at night in their bedroom to an odor. What we did is we had a little machine by their bedside putting out either a citrus or a balsam odor for 2 weeks. And what we showed was we tested their thresholds for that odor weekly before they were ever exposed, and then at the 1 week interval and the 2 week interval, and then after we took it away. And just to show you here, this is their threshold for the odor that they were exposed to. So as the graph goes up, that means they're becoming less sensitive to the odor. So they started out with both odorants at about the same threshold level. And then they started to become less sensitive to the odor that they were exposed to every night for 2 weeks. They became more sensitive to the odor that they weren't exposed to. After we took it away, they recovered their sensitivity, but it actually took almost two weeks for them to get back to the same level of sensitivity after we removed the odor. And it turns out that this increase in sensitivity that we see is actually found in females but not males. So here we tested 5 males and 5 females on 8 sessions to 2 different thresholds, 2 different odors. And what you can see here is an incredible increase in sensitivity. This is for this odor that they were being regularly exposed to in threshold testing called citralva. It's a citrus odor, monomolecular compound. And the red represents the responses of females becoming way more sensitive, orders of magnitude more sensitive. And here we have the guys up here. They learn a little bit, but they're just not showing the same level of sensitivity. They basically, when I brought this graph home, I put it on the refrigerator door, and I told my husband, I decide when the milk has gone sour from now on. And personally, it's not, what we find is this is hormonally gated. This works for, this happens in women that are post-pubescent and premenopausal. And we think it probably has some role in, the protection of, people when they might be pregnant and avoiding compounds that might be dangerous, just like pregnant women often become hypersensitive to odors. But nevertheless, what this means is that you could have a female that is actually complaining. You may be reducing the odor emissions from your site, but you may have someone in the community who is becoming more and more sensitive to that odor simply as a consequence of smelling it intermittently at low levels. The other thing I want to talk about is these two processes in perception. Mostly what I've been talking about up till now is what we call bottom up, what comes from the sensory stimulus, the odor itself, the molecules itself. The other part of odor perception is what our brain does with that information. As Craig said, when you see flowers, you expect to smell flowers. When you see something that looks like it might generate a nasty odor, you kind of expect to smell a nasty odor. And that's a significant driver of what we smell and how we respond to it. So a very, very famous psychologist said this a long time ago, that the general law of perception, part of what we perceive comes through our senses from the object before us. In other words, the strength of the odor, the quality of the odor, whatever. But the other part comes out of our own head. It's how we interpret what we're smelling. And that is a huge driver of complaints in the environment for people. This was a study done by a colleague a long time ago, but it still holds up today. Basically, he was looking at how people who were worried about an odor hazard, how they expressed that in terms of their symptom responses. This was a hazardous waste site that occasionally generated odors, but he surveyed the community around there, and what he found was that, the more frequently they smelled the odor and the more worried they were about what they were smelling, that related to the prevalence of headaches in this population. So basically, everybody was it just didn't matter how close or how far they were from the facility. It was the frequency of smelling an odor and their worry about the odor that caused them to report more headaches in response to that odor. We took this into the laboratory and actually did a study where we intentionally misled people about the nature of what they were smelling. We gave people the same chemical, and we did this with a number of different chemicals over different studies. And in one case, we told them it was a botanical from a rainforest plant. In another case, another group of people, we told them simply that it was a standard olfactory test odor. And in the third case, we told them it was an industrial solvent used to degrease machinery and that it, sometimes caused people to have some symptoms. And basically what we did is we sat them in a chamber for about 20 or 30 minutes, and we asked them to rate the odor intensity over time and then asked them about their symptom complaints before and after. And as you can see here on the right, people in the negative condition reported significantly more nose, eye, and throat irritation than did people in the positive condition, or even in the neutral condition where we really didn't tell them anything about the nature of the odorant. And they also thought that it was more positive, pleasant, relaxing, and healthy in the positive condition than in the negative condition. So here, same exact chemicals, same intensity, same concentration in the chamber, different groups of people respond very differently based on how we characterize the odor. We also did this with a drama student from the university, where we had a Confederate essentially come in and sit in the room with another person who was actually the true participant in the study. And instead of me characterizing the odor, this Confederate was given a script to either talk about how much better they felt, how much more decongested they felt, how much better they were breathing, or they started complaining about symptoms. And the real measure in this case was what the real participant was telling us anonymously, because they were just responding on their computer. And as you can see here, in the positive, negative, and neutral condition, they reported much more eye irritation, nose irritation, a little bit more nausea, drowsiness. They even reported coughing more, even though they never coughed. They just thought they did because the Confederate actually coughed. So in terms of social interactions in an environment, you can see how people complaining about symptoms or problems with an odor-emitting facility can actually induce their neighbors or people that they're talking to actually feel similarly. Craig also talked about the idea that we smell what we see. We also looked at this in a number of examples in the laboratory, and here we took a rather unpleasant odor, 3-methyl-indole, which is one of the main constituents of a fecal odor. And among other odors and other visual scenarios, we showed this odor to people once when they were looking at a porta-john, and once when they were looking at a barnyard image of fluffy white sheep. And probably not surprisingly, when we asked them to rate how strong it was and how pleasant it was, even though it's not a very pleasant odor, it was rated less intense when they looked at the sheep and less unpleasant when they looked at the sheep. So basically, they were responding to the visual input in terms of how they were rating the actual odor. So this goes back to seeing a plant that actually has the potential to generate odor. If you have a nice tree buffer there and they can't see what you're doing, that's one thing. This is incredibly a problem for landfills, which usually rise much higher in the environment than any of their surrounding features and are very visible, but certainly it can be visible in digestion plants and composting facilities as well. And this will actually cause people to have that expectation about odor. And so anything that they do smell then is already biased by what they're seeing and how they're responding to that. So the bottom line is that our perception as humans in response to the type of odors that can be generated from anaerobic digestion or compost facilities depends on multiple factors. Obviously, there are factors in terms of the chemicals themselves, the odor and concentration and its quality. Also, the temporal pattern of exposure. How long does it last? Is it frequent, infrequent? And then individual sensitivity, the variation due to either genetics, age, gender, or disease states. But finally, I think we really need to come down to understanding the role of these expectations, beliefs, and attitudes about the type of odors that can be generated from these facilities and the social context in which people become active about wanting to do something about these odors. The human olfactory system is very unique in that it's optimized to respond very quickly, but also very much capable of arousing individuals to the presence of even low-level chemicals in our environment, particularly ones that are novel, unexpected, or potentially associated with some hazard. Humans appear to be highly sensitive to the odors of organic decomposition, and there's probably a very good reason for that, that we can detect them and find them to be unpleasant at very low concentrations. They can then elicit annoyance and symptoms, but the symptoms are largely, as I said, the true irritation concentrations are likely not to be achieved anywhere off-site, maybe not even on-site, except for maybe certain levels of ammonia in areas. But it's this anxiety about our response to a perceived toxin despite assurances of safety. Because when you think about it, you can't see odors. All you can do is trust your nose, which we have very little confidence in. It's not a good detector in terms of how much odor is in the air, how much chemical is in the air. And so resolving these odor issues is not easy, obviously. We've heard about phyto investigations, frequency, intensity, duration, which all can be measured objectively. Offensiveness is a much more subjective character. Obviously, that is the first step in any odor investigation, but it's also important to keep in mind that the psychological and social factors can also be driving the adverse response, despite all of your efforts to minimize emissions and odorous transport off the site. And if there's any time, I'll be happy to take a few questions. If not, we can just move on. Okay. Questions? Yes.
Repeat the question. You mentioned earlier about measuring, and you know how the European Union measurement protocol is connected? They just put laws to Europe, the EU, to manage odors
Yeah, the question is how the EU manages odors. We actually use the EU odor protocol in our laboratory with olfactometry. Generally, what they do is they take samples from the environment and they bring them back to the laboratory where they're diluted using an olfactometer with clean air. The European Union regulations call for a panel of no fewer than 4 individuals. Generally, it's 6. We have a 6-person olfactometer in our laboratory that was built in Germany to those standards, and that's how it's done. And you ramp up the odor from the highest dilution, in other words, the very lowest odor that might be smelled, to the point where everyone on the panel has detected it correctly a certain number of times. And that's how they come up with their dilutions to threshold for the odors that are collected. I think there's less use of the nasal ranger in Europe than there is in the US right now. I don't hear a lot of people talking about using it there, but that's obviously a way of collecting that same kind of information in the field, as you'll hear about later.
The lab you mentioned, is that the St. Croix method in the lab?
The St. Croix method in the lab, this is actually the European standard for olfactometry that we're using, which is the 6-person, 4 to 6 people, using an ascending method of limits where you start at the lowest level of odor and you continue to increase it until everyone on the panel has detected it successfully 2 times in a row.
In about mid-2000s, I was on the Air and Waste Management Air Committee, along with Chuck McGinley. And basically, at that point, we had adjusted the ASTM method to basically mimic the European method. And what it really came down to was the old ITRI olfactometer, the presentation rate was much lower. Yeah. Okay, so the question was, were you getting enough of the odor for the people's different noses and how their noses fit together and so on. So the 20 liters a minute from Europe was, at first it was considered, not possible because of the taking samples in 10-liter bags, would you run out of material and so on, but we realized it would work. So basically now, the ASTM method is the European method.
What I was just questioning more was about compliance. This law, and are they taking bag samples for compliant challenge circumstances in Europe? Is that how they're making sure that everybody's compliant? Are they using the ASTM? Is that how they enforce the law?
Can you repeat the question? Sure.
Yeah, the question is, how are they enforcing compliance in the EU, with these bag samples and, other methods of evaluating the dilution to threshold? Yeah, you go ahead talking now. I mean, there is no uniform regulatory body that looks, the Scandinavian states do it a little bit differently than Germany, there's not uniformity, but they are using these bagged samples brought back into the lab where dynamic olfactometry takes place with a panel.
Well, I'm not going to challenge it
Yeah.
because there isn't uniformity. My question is, because they just enacted this law across the EU, I was just wondering if they were getting to uniformity, if they had come to that. So it just got enacted in January. It just began.
Yeah, there are groups that meet regularly and do, sort of round robin olfactometry, from all these EU countries. But as yet, I don't know that there is a single standard that they're all adhering to enforce compliance for the industries.
There were some other questions.
Yes, sir?
How accurate is the science for collecting odorous gases and bringing them to a lab and analyzing them for those particular chemicals that cause specific odors? Is that done with some accuracy, or is it mostly done with human noses?
The question is, how accurate is the ability to collect, odorous materials in the field, bring them to the laboratory and analyze them? Well, when you can certainly take that bag sample and run it through a GCMS and look at the constituents. But if the issue is odor, the nose is the best thing to actually evaluate that, because the constituents aren't going to build, we still don't know enough about the way the brain and the nose actually perceives mixtures to know how that mixture is going to be perceived as an odor of a particular intensity. That part of the equation is still very rudimentary. So you can indeed we do a lot of GC olfactometry at Monell, where you take the sample and you feed it through a GC, and part of it is going to the detector, whatever, FID, FPD, whatever you need, and the other part of the sample is being separated and coming out to a port where the person can sit there and smell, and that can help you understand what are the most malodorous components in that, because you can then tie that retention time to a specific chemical. Sampling in the lab always has some inherent problems because you have to bring your sample back very quickly and analyze it. Even with the most inert of materials, Tedlar bags and some other materials that we use, you can still have considerable adsorption on the surface that might make you miss one or two compounds in the mixture. So it's a trade-off between doing the field olfactometry, doing the GC olfactometry, and then doing the standard olfactometry panel. It's as good as it gets right now. People are still working on electronic noses. And quite honestly, we are starting to use DNA-coded nanotubes to actually do the detection, trying to model biologically the human olfactory system with an electronic nose. That's still a long way off, but it's getting better than it was. I mean, the first electronic noses, I think, had maybe 8 or 16 sensors, which doesn't even come close to the complexity of the way the human nose perceives information. But the field is evolving. One more question? Yeah.
Great presentation, by the way, and I love your logo. I don't know if everybody's noticed there's a nose.
Yes. It sits out in front of my building, yes.
Going back to you talked about odor from the porta-john and then having the nice fluffy lambs there. I have a facility, just took over food waste, digesting food waste, long history of odor issues. Am I better to bring some of the local folks who've been highly problematic or found the facility highly problematic? Should I bring them in and show them, look, this is right from the grocery store, and it's this, to give them this feel of, or are they going to look at that and say, yeah, but there's also a bag here that this is the bag that came from my house today, which has the coffee grounds, the lemon peels, and all that other stuff, and yeah, sure, it smells. Am I better to bring them on site, or am I better to not?
My experience is that I think understanding what the source of the odors are is important for people. I had people complaining about a medical waste treatment facility, and they imagined all sorts of horrific things were being, incinerated there, until one woman brought home a pizza and let it sit for a while. And when she opened the cardboard box, she smelled that odor and finally realized, oh, it's not human parts that are being incinerated. It's like cardboard, paper materials and stuff. So I think that level of understanding can actually be very helpful, because in the absence of that, people imagine all sorts of things as the source of that odor. Okay. Thank you very much.
Thank you very much. Bernie, you should paint lambs on the side of that digester.